Hello everyone, we are Team Sweet Success. So the motivation behind our project is India, the world's second largest sugarcane producer, faces a compensation crisis due to inaccurate sucrose measurement, resulting in losses for farmers and sugar mills. Current methods rely on expensive lab equipment inaccessible to farmers, leading to delayed and unfair compensation. So we did some background research and found out that current sugarcane analysis methods are costly, lab dependent and impractical for field use. Large instruments, skilled professionals and expensive chemicals hinder accessibility and fairness in compensation for farmers. Also, sugarcane has three main components: fructose, glucose, and sucrose. As it grows, glucose is used for its development. Sucrose synthase converts excess glucose and fructose into sucrose, which increases as the plant matures. In premature sugarcane, sucrose is 15 millimolar, while glucose and fructose are 110 millimolar. At maturity, sucrose rises to 0.5 molar, and glucose and fructose drops to 0.05 molars, signaling the ideal harvest time. Post maturity, invertase breaks down sucrose, helping the plant survival. Farmers often lack awareness, leading to premature or delayed harvesting, affecting sugar yield and causing losses for sugar mills. So, our problem statement is: how can we create a low cost, easy to use solution for fast and accurate analysis of sucrose content in sugarcane crops for optimal sugar yield? Now that we already know the problem statement, let's have a look at the solution that we have modeled. Our sensor works by taking a sugarcane juice sample, diluting it, and applying it to a special paper. This causes a color change. Using a mobile app, farmers can scan the paper to analyze the color, getting insights into sugarcane quality and sucrose content. The process involves converting sucrose and glucose and fructose, followed by glucose turning into red color dye. This helps farmers make informed decisions on when to harvest the optimal yield.
Last semester, we focused on biochemistry to be used for this sensor. We want to find the sucrose concentration in the sugarcane juice. Invert is an enzyme that can break down sucrose into glucose and fructose. Glucose further converted into gluconic acid and produces H2O2 in the presence of glucose oxidase. H2O2 further react with 4-amino antipyrine and phenol 4-sulfonic acid to produce quinoline dye in the presence of peroxidase enzyme. The quinoline dye is pink in color. This is what will happen to sucrose in the juice, and only for sucrose we need invertase enzyme. However, there is already glucose available in the juice. Let's call it free glucose. Free glucose can also produce pink color dyes in the same manner, but this time we do not need any invertase for this reaction. Now we have quinoline dye coming from the glucose which is derived from the sucrose due to invertase. Additionally, we have quinoline dye coming from the free glucose in the same reaction. Let's call it color produced by total glucose. We can perform another set of reaction without any invertase will only result in free glucose producing the color in the reaction. By subtracting the dye intensity produced due to the free glucose from the total dye intensity produced by the total glucose, and that's how we can determine the amount of sucrose present in the juice. We designed the protocol and conducted a numerous trial and error method to produce the color from the glucose. These are the reagent and their stock concentration to be used in the reaction. We tried plotting the standard curve and got these results. Then we did the optimization for time and found 10 to 12 minutes is the optimal time for taking the absorbance value in the ELISA reader. We also need invertage in the reaction to convert sucrose into glucose and fructose. We conducted codon optimization, cloning and protein expression in the E. coli bacteria and obtained small scale purification. However, we tried large scale purification. There were no protein visible on the gel. As a result, we ordered invertage enzyme from Sigma.
Now, talking about the work done this semester. First, let's talk about the mistake we made last semester. The error we encountered involved discrepancies between the stock concentration and the working concentration of glucose. Initially, we plotted these concentrations against the optical density values of the reaction. Upon closer examination, we discovered that the actual working concentration was 10 times lower than what we had initially assumed. based on the stock concentration. This realization highlighted a significant oversight in our experimental setup, prompting us to correct the discrepancy and proceed with more accurate measurements moving forward. We rectified this mistake and modified the protocol as shown in the table. We prepared a master mix for 10 reactions using glucose oxidase at a working concentration of 1 micromolar per oxidase at 200 nanomolar, 4 amino antipyrine at 0.4 millimolar, and PS at 25 millimolar. The total volume of the master mix was 900 microliter. We then added this master mix to 8 different tubes, 90 microliter of volume in each tube, varying the substrate glucose concentration in each tube. However, we observed that the reaction range decreased after rectifying the mistake. Then we also changed the molarity of the sodium phosphate buffer in the reaction but found that there was no change in the results upon altering the concentration of the sodium phosphate buffer. Initially, in the last semester, we were using the buffer at 700 millimolar, but according to literature and papers, it should have been used at 100 millimolar. We made this change as well, but there was no significant difference in the results. We are currently using 100 millimolar concentration, and the figure also shows that there is almost no difference in the results, although we use different concentration of sodium phosphate buffer in this reaction.
Moving on the further on optimization steps, we attempted to optimize the glucose oxidase enzyme in the reaction. We found that the concentration of glucose oxidase at 300 nanomolar is optimal. Previously we were using at 1000 nanomolar. We conducted the reaction and we used the glucose oxidase at 10 nanomolar. There was no color in the reaction. At 100 nanomolar, there was color after 30 minutes. Therefore, the 300 nanomolar is optimal and providing the better R square value in 10 minutes. Using a higher concentration of enzyme will decrease the time but will increase the cost of product, which we want to decrease, because farmer can wait for few minutes more but will not pay much. Next, we try to optimize the invertage enzyme in the reaction, how much invertage to be used in the reaction, and we found that the 300 nanomolar is the best concentration to be used. Using 1000 nanomolar resulted in the reaction of 8 minutes, reaction time to be 8 minutes, while 300 nanomolar took 10 minutes and 100 nanomolar took 13 minutes. All these concentration provided good R square values, and the time difference can be observed in 96 well plate when analyzing the color intensity. We have some option for binding our protein for surface of the paper. The best choice turned out to be PVDF due to its superior protein binding capacity. PVDF has a protein binding capacity of 170 to 200 microgram per centimeter square, while nitrocellulose has protein binding capacity of 80 to 100 microgram per centimeter square. Hence, we choose PVDF paper to proceed further to prevent our sample from sampling out and potentially disturbing the chemistry. It is essential to create a hydrophobic surface on the paper. The hydrophobic property can prevent seepage. To achieve a hydrophobic region on paper, we can use a polylactic acid solution by dissolving polylactic in dichloromethane, which is then poured over the screen. The creation of hydrophobic layer is crucial to retain the enzyme and maintain sample concentration. For the milestone and timeline, what till now what we have achieved and what we are going to do next. We have already determined the chemistry to be used in the sensor. Specifically, we are using quad pod assay. The concentration of reagent required for the reaction have been already determined. Currently, we are conducting two assays, one with invertage, one without invertage. Standardization of these assays has been completed and we are focusing on optimizing the assay parameters. We have already optimized the buffer concentration, glucose oxidase, invertage, peroxidase, and reaction time. Although we attempted to produce invertage in our lab, we encountered difficulties when scaling it up for large scale purification and production. Our next step will be to test the assay using real life sample, that is sugarcane juice. Once we confirm that it works effectively with sugarcane juice, we will proceed with lyophilizing the assay on the paper membrane. We have already selected the membrane to be used. By the end of April, we plan to begin fabricating the channel on paper and complete our project.
Now talking about the contributions of each member. Since this is a research project, we have not divided our work. We have been working together, we perform the experiments together, and we take care of the data together so that no one misses anything and we could be more productive in terms of the work done. Thank you so much.